Berglund. I'm here to talk to you about the experiments we did on the organism Cyanophora Paradoxa. This organism Cyanophora Paradoxa is of interest scientifically because it is one of the earliest discovered examples of endosymbiosis, a characteristic in which a smaller organism is absorbed by a larger organism in a symbiotic relationship. Another example of this is the mitochondria within our own human cells. Originally, it is posited that these were their own organisms but were absorbed over time into eukaryotic cells to aid in the production of energy. The cyanelles of Cyanophora Paradoxa function in much the same way in that they are similar to chloroplasts. They conduct photosynthesis and provide energy to the cyanophora host cell. Our experiment consisted of measuring the velocity of Cyanophora Paradoxa cells at given light intensities. Our hypothesis is that since the cyanelles will have more light energy at higher light intensities to conduct photosynthesis with, the Cyanophora Paradoxa cells will move faster, just simply because they are producing more energy. Our procedure for this consisted of the following. First, we obtain the samples of Cyanophora Paradoxa and put them onto different plates. Next, we set the microscope light to a light intensity and measure the light intensity using ImageJ software. Following this, we rigged up a microscope cover to block external light, excluding external light with the exception of the microscope light itself from entering the petri dish. This allowed us to get a baseline intensity reading that we used, and we used a similar procedure to obtain the brightness for all following experiments. We then took images, several different videos of Cyanophora Paradoxa cells moving in the petri dish. We used video point software to capture this and then track their velocities instantaneously. We repeated this 21 times over 3 different light intensities. To analyze the data, we used a two-sample student's t-test, which essentially measures the relatedness of each light intensity to the other. We found no significant difference between the 3 different light intensities, which suggests that the light intensity does not, in fact, affect the velocity of Cyanophora Paradoxa.